Osmosis is a very important process in biology that allows water to pass from inside a cell to outside and from outside to inside the cell. Osmosis occurs with solutions. When you have solutions separated by a semi-permeable membrane, water and small molecules like oxygen or carbon dioxide can pass through the membrane, but ions like sodium or phosphate and large molecules like glucose cannot pass through. What happens in osmosis is water moves through the semi-permeable membrane from where the solutes are less concentrated to where they are more concentrated. In other words, water moves to try and dilute down the more concentrated solute. So what we see in this picture is a diagram that shows water, which are the little blue spheres, moving from where the solute, the red spheres, are more dilute to where the solute, the red spheres, are more concentrated. So basically the water is trying to dilute down the more concentrated solute. And if you look at the picture over to the right, you can see that what will happen with time is water will move from an area where there is less solute to where there is more solute, and it will actually cause the level of the water on the side where there is more solute, the red balls, to increase. And this will cause a pressure against the membrane. This is called osmotic pressure. To stop this flow, you actually can stop it, but it requires energy, and you have to press down on that solution that has the more solute molecules in it. And if you press down hard enough, you can stop the water flowing into the solution that has more solute particles, and you can even reverse that and cause water to squeeze out of where the solute particles are. Basically, water is trying to crowd around all those solute particles and dilute them down. And if you press on it hard enough, you're going to make it so that they can't push in. To purify ocean water so it's drinkable, they will use this process called reverse osmosis by taking the seawater and pressing hard on it against a semi-permeable membrane, and pure water will flow through the membrane if you get the pressure high enough. Now in cells, you will find that sometimes you can put a cell into a solution that has fewer solute particles. In fact, the solution would be less concentrated on the outside than it is on the inside. Well, this is called, the solution around the cell when it's less concentrated is called a hypotonic solution. Hypo means low, so it's a low concentration out there. So the water is going to try to flow into the cell to dilute down the more concentrated solutes inside the cell. What happens is the cell will be absorbing all that water as the water tries to dilute down the inside of the cell, and it will eventually swell up to the point that it bursts. With red blood cells, this is called hemolysis. Hemo for the red blood cell, and then lysis means cutting. So the red blood cells burst open and die. If the solution that you put around the cell has more solute particles in it, and that means the solution outside the cell is more concentrated than the solution inside the cell, then the solution is called hypertonic. Hyper means too much. That means there's too many solute particles outside the cell. So the cell has a more dilute solution inside. The water is going to flow out and try to dilute down the more concentrated solution outside. What happens if this is a red blood cell is the water will flow out and that little red blood cell will start to shrivel up. That's called crenation. If the concentration of the solution inside the cell is the same as the concentration of the solution outside the cell, this is called isotonic. So, solutions that are isotonic to the body are a 0.92% sodium chloride solution or a 5% glucose solution. So when you put body cells, like red blood cells, in those solutions, they neither swell or they neither shrivel up. And so then the red blood cells would remain healthy. And this is going to be really important to those of you going into nursing, because when you're giving a solution to a person, you want to, an IV solution to a person, you want to make sure that you don't cause their red blood cells either to burst or to shrivel up. So this whole idea of isotonic solutions is very, very important. Now, let's see if you understand this whole idea of hypertonic, hypotonic, or isotonic. Remembering that the concentration of particles in a cell, that would be the solute particles in a cell, is about the same as in a 5% glucose solution. Describe each of the concentrations of these solutions. Pause the lecture, answer the problems, and then you check your answers when you resume. Well, because 5% glucose has the same concentration as the concentration of solute particles in a cell, well then that would be isotonic. When there's more solute particles in the solution, as in a 10% solution, there's too much. So that's called hypertonic. When it's 1% glucose, there's too few solute particles in the solution, so that is called hypotonic. It's too low. Now do these problems. Try and decide which way will water flow under these conditions. Pause, do these problems, and then press resume to check your answers. When a strawberry is placed in distilled water, the distilled water is hypotonic because it has too few solute particles. This means there is too few solute particles compared to the concentration of solute inside the strawberry. The more dilute solution, the distilled water, is going to have the water particles there flow into the strawberry, and what will happen is the cells will swell up with the distilled water, and they can burst. When you place a red blood cell in 2% sodium chloride, 2% sodium chloride is hypertonic. Hypertonic solutions have too many solute particles in the solution, and so what happens is the water inside the cell is going to flow out and to try to dilute down that 2% sodium chloride solution. So water will flow out of the cells, and you're going to have crenation occur. Be sure to do problems. There are in-text problems to do as you read, and then the end of chapter problems will reinforce your reading. Remember, do not do similar problems one after another, as this only relies on short-term memory and not on understanding. Mix up the problems as you do them, and be sure and do the worksheets that are posted for the chapter. Don't forget to do your online quizzes.